Thanks so much for joining me for another edition of Parker Biology. Today we'll be looking at how eukaryotes regulate gene expression. All organisms need to regulate which genes are expressed at any given time. It's important that organisms are able to turn genes on and off in response to signals from their external and internal environments. Also, in the case of multicellular organisms, regulation is important for cell specialization. Each type of cell has a distinct role, and in order to perform its role, each cell must only express the necessary genes. Due to the fact that we started as just one cell, the fertilized egg or zygote, almost all of our cells are genetically identical within a given organism. So when we think about the fact that our cells are so specialized, skin cells do what skin cells need to do, and heart cells do what heart cells need to do. These differences result from differential gene expression, meaning that different genes are expressed by cells, even though they all have the same instruction manual. This expression can be regulated at many stages, ranging from transcription, post-transcriptional modifications, and through translation. Anytime we see abnormalities in gene expression, too much of a protein being made or too little, we can see disease results, including cancer. This diagram reminds us of the huge process of gene expression, all the way starting in the nucleus with the chromatin, ending up with functional proteins. And what we'll see is that at each of the steps along the way, regulation can tell the cell either yes, make this protein, or no, the cell isn't meant to have this protein right now. We're going to use this diagram as a roadmap today and keep coming back to it to pinpoint all of the spots that regulation can occur. Remember that the structural organization of chromatin not only helps the cell's DNA fit inside the nucleus, it keeps it nice and compact, but it also helps regulate gene expression. If the cell's DNA is so highly packed or in the heterochromatin form, remember that the cell's machinery, specifically transcription machinery, won't be able to reach that highly coiled heterochromatin. On the other hand, the loosely packed euchromatin is more readily accessible to transcription machinery. We've mentioned the fact that highly packed heterochromatin is harder to read, whereas the loosely packed euchromatin is much easier to access and more likely to be expressed. But now we're going to look at a couple of chemical modifications to the actual histones in the DNA that can influence the structure and therefore the ability to have the genes expressed. Examples of these modifications include acetylation and methylation. In histone acetylation, acetyl groups are attached to lysines on the histone tails. The lysines are positively charged, but the addition of acetyl groups neutralizes this charge. This makes adjacent nucleosomes less likely to bind to one another, in turn loosening up the chromatin structure, which promotes transcription initiation. Here we're reminded of the structure of the nucleosome, whereby the histone proteins are wrapped with DNA. And here we can see that the tails are actually exposed. These are available now for chemical modification. Notice that without the addition of acetyl groups, these nucleosomes pack tightly together. But with acetyl groups on the histone tails, we can see that neutralized charge leads to less tightly packed instructions, which makes it accessible and permits transcription. In addition to histones being modified by acetyl groups, histones can also be modified by the addition of methyl groups through histone methylation. This has the opposite effect of acetylation. It actually tightens up the chromosome structure, which makes transcription less likely. Our improved understanding of histone modifications has led to the development of the histone code hypothesis, which proposes that these specific combinations of modifications, along with the order that they occur in, can help determine the structure of chromatin and thereby influence whether or not transcription will occur. In addition to methyl groups getting added to histones, methyl groups can also be added to certain bases of DNA. This is called DNA methylation, and this can cause chromatin to condense, again making transcription and thereby expression less likely. So this methylation is seen in long-term inactivation of genes when cells differentiate. If they're not meant to read a certain portion of instructions, those instructions can be methylated. What's also interesting is that this methylation can be seen passed from generation to generation of cells. These methylation patterns can be passed on and in fact impact future generations of cells. Even though these modifications do not alter the actual DNA sequence, they can be passed on to future generations. When we talk about inheritance of traits, not directly involving nucleotide sequences, we refer to this as epigenetic inheritance. Scientists are learning more and more about the importance of epigenetic information in regulating gene expression. Epigenetic variations might help explain why one identical twin might end up with a genetic disease like schizophrenia, but the other does not, even though they have identical genomes. Other epigenetic markers, like abnormal methylation, can also be linked to disorders like cancer. Clearly, the packaging of chromatin is going to have a huge impact on whether or not the transcription machinery can access instructions. Once the chromatin is optimally modified, transcription initiation will be the next point that we look at in control of eukaryotic gene expression. Transcription can be regulated by control elements and transcription factors. Control elements are segments of DNA that don't actually code for protein, but serve as binding site for transcription factors, and this helps regulate whether or not transcription occurs. These control elements are located in a region that we call upstream, meaning in front of the promoter, and we can describe them as proximal if they're located close to the promoter, or distal if they're located further upstream from the promoter region. Remember that in eukaryotes, RNA polymerase cannot simply bind to DNA and start transcribing. Instead, RNA polymerase requires certain proteins called transcription factors. Now we're going to examine how control elements can interact with specific transcription factors to enhance transcription. Here we see a cluster of 3 distal control elements grouped in what we call an enhancer region. Activator proteins bind to these enhancers, and when activators do bind to enhancers, this should enhance or promote transcription. How does that happen? With the activators bound, a DNA bending protein will bind and literally bend this DNA, which helps bring these bound activators in closer proximity to the promoter region. The activators are then able to bind nearby mediator proteins along with transcription factors. Because of this bending, because of this proximity to the promoter region, these activators bound to the transcription factors are now bringing these transcription factors in contact with the promoter, allowing RNA polymerase to come in, thus forming the transcription initiation complex. It seems strange that an enhancer region so far from the promoter could impact activity at this region, but because of the DNA bending, these transcription factors are able to be brought in and transcription will ensue. Here we can see how this type of regulation can enable cells to be highly specialized. For example, both liver cells and the lens cells of your eyes have the genes that would help you make albumin and crystalline. However, we only see the liver cells make albumin, and we only see the lens cells make the crystalline. Even though both types of cells have the potential to make both proteins, they have different activators available. Here in the liver cell, we see activators that bring about a bending due to this enhancer region that will allow for albumin genes to be expressed. However, over in the lens cell, there are not the right activators available to make the albumin. They've got the correct activators to bring about crystalline expression. So depending on which activators are there will dictate how the DNA bends and which gene is actually read. While regulation of transcription is a great way for cells to decide which genes they'll express, transcription alone does not account for gene expression. Remember that truly expressing a gene is reading the DNA, converting it to RNA, and then building proteins. So the expression of a protein coding gene is ultimately measured by how much functional protein the cell makes. There are various regulatory options for the cell after transcription, which allows the cell to fine-tune expression and respond quickly to any environmental changes. They shouldn't make a protein if they don't need the protein. Remember that following transcription, the RNA isn't quite ready to leave the nucleus just yet. Eukaryotic RNA must be processed. This includes addition of the 5' cap, the poly A tail, along with RNA splicing, where the introns need to be cut out and the exons need to fuse together. A key step in gene regulation is deciding which portions will be treated as coding regions or exons, which portions will actually go on to be exported and translated. The fact is that we can splice in different patterns, and this is called alternative RNA splicing, which means even though we have the same general piece of pre-mRNA, depending on how it's cut, could result in a variety of different polypeptide chains. For example, here we see the troponin T gene. Notice that different splicing can result in different RNA. Here we can see that region 4 has been excised, whereas another pattern leaves region 3 excised. So depending on which regions get to stay is going to determine which kind of polypeptide chain we create. So eukaryotes can regulate how their DNA is packaged, whether or not transcription occurs, and the processing of the RNA. Finally, once the RNA comes out into the cytoplasm, it doesn't get to stay forever. The lifespan of messenger RNA is key in determining how much of that message is read and how much of that protein is made. For example, if that messenger RNA is degraded within minutes, not very much polypeptide will result. On the other hand, if that messenger RNA gets to stay around for days or a week, a lot more of the protein will be synthesized. So the lifespan is absolutely vital in determining how much protein will actually result. Hopefully this video has helped you better understand the initial points of control of eukaryotic gene regulation. We've seen how DNA packaging, transcription, and RNA processing can be regulated, along with how the lifespan of mRNA can impact gene expression. Please join me for the next edition of Parker Biology, where we'll look at what else happens out in the cytoplasm with regard to eukaryotic gene expression, with translation, protein modification, and degradation.